So the title of my talk is Origins of Biological Complexity, an appeal to intuition. You'll see what I mean by that as we get further into the talk. So to start off with, I think it would probably be best to go ahead and describe what I mean by biological complexity, right? So here is an encyclopedic definition of biological complexity. So this refers to a measure of the complication of a biological organism that is directly related to that organism's ability to successfully function in a complex environment. And after reading that, you might think to yourself, I'm not sure how much that helped me, quite honestly. So let me unpack that statement a little bit. And let's just do this. Let's think about cells from this standpoint. So cells are made up of parts. So what you're looking at here, actually, let me go ahead. Yeah, I think this will be fine. So what you're looking at here is an image of a cell, or many cells, actually. This is an image from a fluorescent microscope. And what you see highlighted here are some of the parts of a cell, right? So in green and red are the cytoskeletal elements of the cell. So cells actually have skeletons as well. They're made of proteins. And then in blue, you would see the nucleus. So this is a fraction of the number of parts of the cell, but cells are made up of parts. And biochemists think of the parts as being either macromolecules. So there are four classes of what we call macromolecules. And macromolecules are large molecules. Proteins are macromolecules. Nucleic acids, which are DNA and RNA, are macromolecules. Lipids are macromolecules, and the carbohydrates are molecules. Okay, so cells are made up of parts. Right now, those parts are going to then participate with one another. They're going to interact with one another to carry out the various processes of the cell. Some of these processes you may have heard of. So for instance, metabolism is a cellular process that takes nutrients and harvests energy from them. For metabolism to occur, there has to be a large number of the parts interacting in an orchestrated and coordinated way to harvest that energy and then to store that energy is something like ATP, which we'll kind of talk about later on as well. And so cells allow parts to then carry out processes, and processes are going to be driven in a certain direction by driving forces. We won't spend a lot of time talking about driving forces, but the cell needs energy to carry out its processes. So that's part of a cell. And then finally, and very importantly, cells also require a tremendous amount of information that then really directs the production of the parts and the processes that occur within a cell. And the information within a cell is stored primarily in the genetic material, specifically DNA, which is found primarily in the nucleus. Okay. Now, let's draw an analogy for you that might, help you connect with what I just said about cells, parts, processes, driving forces, information, and the biological complexity. And let's put up another image here. So this is one I think that most of us are familiar with. In fact, some of you may actually drive a GMC Sierra. So this is a pickup. It's a very nice pickup. And when we think about an automobile, or we think about a vehicle or a pickup, it's made up of parts, as we know, right? There are parts in the engine, there are parts in your cab, there are parts, all sorts of parts. In fact, if you were to Google, and I did this, if you Google how many parts are in a GMC Sierra, it says, Google says lots. Okay, so there are apparently a lot of parts in a GMC Sierra, but we know this, that those parts are going to then interact with one another in particular ways to carry out the processes that allow the pickup to drive down the road. Okay, and to do lots of other things like open your window or drive the windshield wipers or whatever, right? And then there are driving forces. Pickups have, of course, a battery, and then we fill them with gasoline so that they can go down the road. And then, certainly newer vehicles, have entire displays that have software programs that are running those displays. All sorts of information is required to then allow for the pickup to go down the road. So if you're to come back to our description here of biological complexity, we could make this automobile complexity. And the automobile complexity refers to the measure of the complication of an automobile that is directly related to that automobile's ability to successfully function in a complex environment. In other words, there are a certain level of complication that has to be present for the pickup to function the way we expect a pickup to function. And so that is also true then of a cell and a biological organism. There has to be a certain level of complication for it to operate in the way that we expect it to operate. And that is to, of course, sustain life and biological life. Okay, so that's biological complexity. Now, when we think about the origins of biological complexity, what we're thinking about here is just really two main options that we have presented to us that have been debated for centuries and centuries. And one of those options, in terms of how all of this came about, is just an undirected material process. And this says that there's time plus energy plus chance is a sufficient explanation to describe how all of biological complexity and all biological organisms came into existence. Right? The other, of course, option would be that there is a material process, but there's an engineer behind the material process and an intelligent agent. Okay, and it is this second part that I want to spend some time thinking about. And if it were true, and if it is true, that there's an intelligent agent or an engineer behind biological complexities and the origins of biological complexities, then we should be able to detect that. We should be able to recognize and see that the marks of that engineering. And really, we're going to address this question in the first part of my talk. Are there then features of a biological organism that exhibit the marks of intelligence? And so that's what we're going to be thinking about here. And this can be a very complex answer. We give a very complex answer, or we can give one that, in my opinion, you can just draw on a person's intuition. Okay, and that's what I'm going to try and do, is draw on your intuition to help you recognize that there are features of biological organisms that exhibit the marks of intelligence. Okay, now to answer this question, let's do this first. Can we, as human beings, recognize when something's engineered, recognize when something's been designed? Okay, so let me ask you to do this with me, okay? So let's pretend that you step into a room, and in that room, you see a lotto ball machine, such as being depicted here in our figure, okay? But in the hopper of this lotto ball machine, instead of having balls that have numbers on them, there are lots of balls that have letters on them, okay? And in the chute of our lotto ball machine is balls that have letters that make up this sequence of letters, okay? And starting with L and ending with I, and there's 150 characters in this sequence. Now, go ahead and study that sequence, and what I want you to ask yourself is, do you think that that came about due to chance? Or is there something else behind it? Was it designed? Okay, now, as you consider that, let me go to my next slide, and let's just think about it from a chance standpoint, okay, and the probabilities of getting that sequence of letters. Okay, so this sequence of letters, like I said, is 150 characters long. And the probability of getting that sequence is 1 in 26 raised to the 150th power. Okay, where do I come up with that number? Well, let's think about this. Your chance of getting an L in the first position is 1 in 26, right? Because there are 26 letters in the English alphabet. And so then we have a 1 in 26 chance of getting the L in that first position, okay? Likewise, we have a 1 in 26 chance of getting an X in the second position. Okay, now these probabilities are compounding, so we would say that we have essentially a 1 in 26 raised to the second power of getting LX as our sequence. Okay, take that across all 150 characters, and we come to this probability. To get that sequence of letters is going to be a 1 in 26 raised to the 150th. Okay, I am a scientist, so I like to think in scientific notation. So I converted this to base 10, and so there's a 1 in 10 to the 212th chance of getting that sequence of letters. Not very probable. Okay, so now before we go on, I want to think about that number because it'll be important for us later on. That is a big number. Okay. Let me try and illustrate to you how big this number is. Okay. So when we think about numbers like this, let's first of all just think about a million. Most of us have a context for a million because if we put a dollar sign in front of a million, we think, well, that's a lot of money. Okay. And we have some concept of what that means. A million is 10 raised to the sixth. Okay. A billion, now put a dollar sign in front of that. That's a big, big number, right? That's 10 to the ninth. A trillion is like a very, very large amount of money, very hard for us to conceive of, but a trillion is 10 to the 12th. Now, that's 200 orders of magnitude smaller than our number right here. But it's hard to even conceive of how big this number is. So let me give you some idea of how big this number is. Our current national debt is estimated to be at about $35 trillion. Scary number, right? So if we started to pay off the national debt at a rate of $10,000 a second, it'll take about 104 years to pay off the national debt. Now, understand that number, 35 trillion, is 35 times 10 to the 12th. $10,000 a second, 104 years. That gives you an idea how big this number is. So then put that in contrast to how big this number is. That number is so large. It's almost impossible to conceive of how large it really truly is. So to give you an idea, astrophysicists, and I'm not one of them, so I don't know how they do this, but astrophysicists estimate that the number of atomic particles or atoms within the universe is about 10 to the 80th. So when we think about this, 1 in 10 to the 212th, if you think about your Lotta machine, you're never going to get this sequence again, right? So it's extraordinarily improbable that you would ever get that sequence ever again, okay? But does it bear the marks of intelligence and design? And I would say no, it doesn't. And I think all of you would say no, although you might be sitting there, I see, trying to convince me that it does. No, I'm not trying to convince you that it does. I'm just trying to convince you that it's a very improbable sequence of letters to get, but that doesn't mean that it's been designed, and that doesn't mean that it's been engineered. However, let's do this. Let's look at this sequence of letters, okay? And kind of study that, and look at that, and let me go ahead and help you. I'll go to the next slide here because it might be kind of hard for some of you to see. Let's go to this slide. Okay. So now what I've done is I've bolded and I've underlined aspects of this to separate, right, the text. And what you can see here is that this says what can be known about God is plain to them, for his invisible attributes, namely his eternal power and his divine nature, have been clearly perceived ever since the creation of the world. We recognize this, many of us have probably recognized this as a passage from Romans 1. In fact, Rob Stadler quoted this, okay, at the very end of his talk. So yes, this is, a portion of Romans 1. Now, we automatically look at this and we automatically recognize that it's been designed, it's been engineered. It communicates something to us, and we can point to, at the minimum, the human author in the Apostle Paul. Right now, here's what's interesting about that. This character, or this sequence of characters, is also 150 characters long, which means that it is every bit as improbable to get that as it is my previous sequence of letters. But we automatically assign intelligence to this one and not the other one. Right? And the reason is because this one adheres to the rules of the English language, specifically the grammatical and the spelling rules of the English language, right? So it has purpose, it has the capacity to communicate, it has teleological significance, if you will. okay? And we can recognize this as then being engineered, designed. Now, we can go further. Right? Let's do this. We have two rocks here. Which one's been designed, which one hasn't? I don't have to spend a lot of time with my audience convincing you that one of these is the result of, right, some designer for a specific purpose and a specific intent. It has teleological significance to it, right? That's the arrowhead over on the left. This, probably not. It's just a rock, right? Let me give you one more example. Okay, hunks of metal. We've got two that I think would automatically be recognized by the audience as being designed, having been created, having been engineered with a specific purpose in mind. There's an intent, there's a purpose behind them, and then the other one not. Right? What's interesting about this one, okay, the one on the left, is that this is a dagger that's several thousand years old. Okay, and I actually give an attribute to the Brooklyn Museum because I took this from their website. This is a dagger that was found in an archaeological dig in Egypt. Okay, and it's several thousand years old. Now, what do you think the archaeologists thought when they first discovered this? They didn't sit there and think to themselves, matter plus time plus chance brought this about. They automatically recognized that there was a intelligent mind that produced this dagger, and they then thought of questions like, why did they produce it? What does this tell us about the Egyptians at this point in time? What does it tell us about their technology, right? All these types of things. And understand that those are questions that are being posed by scientists, and they're automatically saying that, well, this is attributable to an intelligent maker, a designer, an engineer. Okay, so all of that, right, to ask this question, can we as human beings recognize design? And I think the conclusion to that is pretty clear. Human beings do have the ability, an innate ability, to recognize design when they encounter it. Okay, in fact, we would say that it's intuitive to us, right? We can look at something and we can intuitively know whether it's been designed or not. Okay, and this is the idea of an appeal to intuition. Now, you might be saying, I thought this talk was about biological complexity, and he's a biochemist. Isn't he a biochemist? Yeah, I'm a biochemist. So let's go on and ask this question. Can we detect, or can design be detected in biological structures? And can we use that intuition, that innate ability to recognize that there is design in biological structures? So what I want to do is give you some examples of biological structures and things that I think intuitively allow us to answer this question. Right? And we're going to talk about proteins. And this is where I'll, Joel Brown, thank you, Joel Brown, for doing what you did. You spent all that time talking about collagen and what collagen is and what proteins are. And you did a lot of really good work for me because I have all this very similar things. I'm not going to talk about collagen, but I want to talk about what proteins are. So as Joel mentioned, proteins are complex macromolecules. So remember, macromolecules are some of the parts that make up our cells. Okay, so these are complex macromolecules comprised of amino acids linked together in specific arrangements to carry out a particular function. Okay, that's what a macromolecule is a complex arrangement of essentially amino acids linked together to carry out a function. Okay, now, an amino acid. Joel did it with a physical object. I'm just going to do it this way. I don't have the physical object. Good job, okay, on that, but something similar here, okay, that we're looking at. You're looking at here an amino acid, a single amino acid. It's got front and center to it a carbon atom, okay, and then it's got over here another carbon atom in green. So each of these spheres represents atoms. And then we've got an oxygen atom, we've got a nitrogen atom, we've got some hydrogen atoms here. Some of the hydrogen atoms were removed for simplicity, okay? But then we've got this here, okay, this gray one, and that is essentially a side chain or an R group. So Joel mentioned that there are 20 different amino acids, and that's right. So there are 20 different amino acids occurring naturally in a protein. And what differentiates those amino acids is the identity of that R group. okay, that side chain. And those side chains have very different physical, structural, and chemical properties. Okay, so some of them are quote unquote hydrophobic, meaning they're water fearing. Okay, some of them are hydrophilic, water loving. Some of them are acidic, some of them are basic, some of them are negatively charged, some of them are positively charged. Right, so there's different chemical and physical makeup of each of these side chains. Okay, now put this in the context then of a protein. Let's go to my next slide here. Okay, so just as Joel mentioned, I'll mention to you as well. So proteins are going to be how we, when we take amino acids and we link them together, right? So here is an amino acid, okay, and we see here is an amino acid here, okay? So you can see the structure here, the structure here. You see the structure here, see that structure here. See the structure here, see that structure here. Okay, so there's one amino acid. Flip it around and you got another amino acid, okay? And then you flip it around again, you got another amino acid, right? So in this, this depiction, we have three amino acids and we would call this a protein, really a peptide, a small protein, okay? And the amino acids are held together by the peptide bond, which is again, something that was talked about in our last talk, okay? The peptide bond is what holds these amino acids together, right? Now, that's just three amino acids making up this particular protein, but that's a really small, what we call protein. Okay, average size of proteins are somewhere around 300 amino acids. Okay, so let me go to here. Okay, and what you're looking at here would be a way that we depict a larger protein. Okay, this one happens to have 260 amino acids in it. And we represent each of the amino acids with a letter, right? So M means methionine, L means leucine, G means glycine, I means isoleucine. Okay, and on down the list, and there will be 20 naturally occurring amino acids, each one with their own chemical appendage that has its own chemical and physical properties like water fearing and water loving and acidic, basic, charged, not charged, those kinds of things. Okay. Right, so that's then a depiction of a much larger protein in these amino acids when then each of them be held together by the peptide bond. Now, some of you might be looking at this, okay, and saying, well, that's a sequence of letters, kind of like what we talked about with our Lotta ball illustration, right? So that's right. This is a sequence of letters that has, of course, a physical end to it in amino acids making up a protein. But here's our question about this, right? So is this sequence of amino acids like our first Lotta ball result or our second one? In other words, can we detect in this some end, some purpose, some teleological significance that would allow us to say, well, this was designed or not, okay? And so let's first of all just address this from this standpoint, okay? It's really improbable that you would get this sequence of amino acids, okay? So talk about the probabilities, just like we talked about the probabilities of the letters in our, our Lotta ball example, okay? In this case, though, we have to say that our base is going to be 20, okay, and we raise that to 260, okay, so there are 20 amino acids, okay, so we have a 1 in 20 chance of methionine being the first, or M being the first amino acid in our protein, okay. We have again, a 1 in 20 chance of a L or leucine being the second amino acid in our protein, right? All the way to 260 characters, right? So to just get ML, it would be 20th raised to the second. To get all of these, it would be 20th raised to the 260th, meaning that, there are, well, 10 raised to the 338 power ways to arrange 20 amino acids in a protein that is 260 amino acids long, okay? Ooh, super big number, right? Super big number. But just like we learned with our, a lot of all examples, just because it's complex, just because it's very improbable that we would get it, doesn't mean that it's designed, right? It doesn't mean that it has the marks of an engineer behind it. Okay, there has to be something else. So what about this sequence of amino acids? We would actually call this a primary structure. Okay, well, this primary structure, if it's made, if we attach all of these amino acids together, we put them into a biologically relevant solvent, that will actually fold up, that protein, okay, will fold up into a structure like this. Right, so in our previous talk, we looked at how collagen has a particular structure and that gives it a particular function. Right, well, we can say the same thing here about this protein. What you were looking at here with this protein, and there's two different ways to depict this, but this protein is a result of this structure here, okay, or this sequence of amino acids, and this protein we call chymotrypsin. And chymotrypsin is a very important digestive enzyme, okay? Now, what you see on your right, probably what the protein really looks like, okay? What you see on the left, we call this the cartoon model, kind of like Mickey Mouse, which has features of a mouse, but not really a mouse. Okay, this has features of a protein, but is not really what a protein probably looks like. Okay, however, I can use this to try and help you understand what it means when I said that the protein folds. Okay, so what you're looking at here in the center, right? What I've colored here in kind of cyan. Okay, same thing over here on the surface representation. But what we see here are these ball and sticks, and these ball and sticks represent those appendages on particular amino acids within our sequence. I say, all right, so this one here would be the amino acid aspartate. This one here would be the amino acid histidine. This one here would be the amino acid serine. Okay, and notice that they're all right next to each other in this structure. Right now, let's go back to our primary structure. Okay, in our primary structure, we can see here, I've colored again in cyan, those three amino acids that are right next to each other in this structure, what we call our tertiary structure or the globular structure. And trying to illustrate what's happening here is that these are a long ways away from each other if you were to just draw out the chain of amino acids, right? But then we let the thing fold up and it forms into this spherical globular structure. And it puts these three amino acids right next to each other. Now, you might think, okay, I think I see what you're saying. So it's kind of like a noodle, you know, when it starts off really long and then we let go of the ends of our noodles and it kind of folds up on itself and forms this ball and it places specific things right next to each other. I hope that's what you took away from that because that's exactly right. Okay, it kind of folds up and puts specific amino acid side chains or R groups right next to each other. Okay, so what does that mean? Well, that means a lot. Okay, so this protein, chymotrypsin, that we see here is an enzyme found in your digestive system. It's produced by your pancreatic cells. Okay, and it allows your body to hydrolyze peptide bonds. So you and I eat protein-rich meals. We should eat protein-rich meals, but we can't absorb the proteins in our meal. We have to break those proteins in our stomach down to, not our stomach, but also our entire GI tract. We have to break those down into individual amino acids, which means we have to break the peptide bonds. Okay, now Joel spent a lot of time talking about peptide bonds and the degradation of collagen and that kind of thing. Well, yeah, those peptide bonds, they're super stable, right? And what you were looking at here, this is a peptide bond, okay? And what's being depicted over here is the hydrolysis of that peptide bond. So we're going down. We'll start here with this is our reactant, and we start here with water, and we go in this direction, and water can break that bond and make two individual amino acids. Okay, we've broke the peptide bond. Now, that takes a long time, hundreds of years, okay, without an enzyme. However, if we have chymotrypsin, it can do it in seconds, right? So chymotrypsin can catalyze the rate of peptide bond hydrolysis millions of fold. Okay, so it can make it super, super fast so that we can then break the protein down into individual amino acids, absorb those amino acids for our health. Okay, and that's what chymotrypsin does. Now, that gets back to our question. Is this more like our first lotto ball sequence or our second lotto ball sequence? Well, it turns out that this is a lot more like our second lotto ball sequence. And the reason is because it produces this that does that. In fact, we can think about this structure as being something like this structure. It has teleological significance, it has purpose, it has intent behind it. It is doing something, right? And these three, the placement of these three side chains is crucial to the function of chymotrypsin. If you were to remove one of these or change their positioning, it will not be able to hydrolyze peptide bond. Similar to if we were to drill a hole in the top of this piston all the way through this, this would cause a malfunction in the engine. So if we were to change one of these three, it would cause a malfunction in chymotrypsin and malfunction in your ability to ingest certain types of protein, and you would feel sick because of that, okay? So very, very interesting when we start to think about this from a ability to detect design in biological structures, okay? Now, the rabbit hole goes deeper. Okay, so let's move beyond this. Hopefully you see what I'm saying here, right? This produces that, okay? Here, which does that, okay? And it has all those features that we intuitively know something about this, okay? But let's go further into this and let's come back to this. And you say, oh, those letters again, the sequence of letters. That's exactly right, the sequence of letters again, right? So here we are. This is our first lotto ball results. And if you were to walk into our room and you saw this lotto ball machine with this sequence of letters on it, you would think to yourself, probably just chance that that happened. However, as you get closer to your lotto ball machine, you see the sticky note. And on the sticky note, there's instructions that tell you how to use a decoding system for this sequence of letters to get a message. And you think, well, that's odd. And so you read through those and you decode it. And it turns out that in that decoding instructions, this sequence of letters actually leads to this sequence of letters, which is, remember, a part of Romans 1. So maybe the decoder said, everywhere you see L, put W. Everywhere you see X, put H. Right now, when you discover that, you think, wow, someone had way too much time on their hands. Okay. But notice what you're saying. Someone, an intelligent being, had too much time on their hands, right? Because we know that codes and then instructions to decode something come from an intelligent mind, an engineer, right? That's something that we intuitively know and recognize. So you think, okay, so why are you telling me that? Well, because this kind of thing happens all the time in cells. There's an entire decoding system. Okay, so let's go here. Now, let's think about this. Come back to, actually, let me go back. We'll go back several slides. Come back to this. Remember I said that this is this is one way out of 1 times 10 to the 338th power, okay, to arrange 260 amino acids. Right? So you would never see it by chance, you would never arrive at this, okay? However, your pancreas makes this all the time, okay? In fact, I venture to say that your pancreas will probably make that 1 trillion times in your lifetime. Okay, maybe more. I don't know. I honestly don't know. I'm just guessing, but it's going to make it over and over and over again. And here's how, and here's why it can do that. The reason is because there's information stored in your cells that tell the cell how to make that protein, right? So if we come down here, this would be our protein sequence, that 260 amino acids that makes chymotrypsin, okay, that we've been talking about. The information to make that is found up here at what we call the DNA level. Okay, and what's shown here in blue would be the DNA sequence, starting with this arrow pointing in that direction and this blunted arrow pointing in the opposite direction. That's what we call a gene. okay? And that's found in our DNA. And there's this process that scientists, biochemists call transcription that takes that gene and converts it to an mRNA sequence. Transcription. This takes a large number of protein machines and other genes to do this, molecular machines to do this. Okay. And then that mRNA sequence can leave the nucleus, go out into another environment in the cell that we call the cytosol, and it will encounter a large number of other machines, RNA and protein machines, and that will go through a process called translation, where it will actually change languages from RNA into the protein. Okay, to understand what we're saying here, this encodes, this DNA encodes this amino acid sequence. Okay, and let me kind of show you what I mean when I say this. Okay, so let's go to my next slide here. We have here, I'm not going to put the entire DNA gene on this slide because I would have to have a font of like 3 to fit it all on here. Okay, so that wouldn't be very fair to all of you. So we'll just have the very beginning of it. But in DNA, this is what the beginning of the protein coding portion of the gene would look like. Okay, and it would have this ATG, and then there would be CTT, and then a GGT, and an ATT. Now you think, what are those? Well, those are nucleotides. Okay, so nucleotides are equivalent to amino acids. Amino acids are the building blocks of proteins. Nucleotides are the building blocks of DNA and RNA. Okay, now there's 20 different amino acids that will make up proteins. There are 5 different nucleotides that will make up DNA and RNA. Okay, A, T, G, and C make up DNA. Right? And we see those right at the beginning, A, T, G, and C. Right? These have different structures and different chemical properties, but they're also very similar to each other, and they're linked together, not through peptide bonds, but something called a phosphodiester bond. Okay, we won't look at that bond. Okay, but they're linked together. Right now, DNA is transcribed to mRNA. The only difference between DNA and mRNA from a nucleotide standpoint is that instead of having T in mRNA, there's U in mRNA. Okay, so AUG instead of ATG. Okay, now there are other differences, structural differences between DNA and mRNA that aren't important for this talk. Okay, but the process here is transcription. Okay, and so transcription really is transcribing the information in DNA to mRNA. Now, we need to then move from here. Now, understand we're only showing a very small portion of the mRNA as well, but this encodes this. Okay, in fact, every 3 letters of the mRNA are called a codon and encodes a single letter or a single amino acid in our protein. Right, so that this is true, that AUG will dictate the first amino acid to be M or methionine. The next 3 letters, the next codon, is CUU, and CUU encodes an L or a leucine. Okay, the next 3 letters, GGU, encode for the glycine. The next 3 letters, AUU, encode an isoleucine, right? So that we can look here, the first 4 amino acids, MLGI, are encoded by the first 12 letters of our mRNA, which corresponds to what's in DNA. Okay, understand that this is a decoding process, and this happens all the time in something that we call gene expression. So if you look at transcription and translation together, we call that quote unquote gene expression, okay? And genes are expressed through this translation process using something called the genetic code, which in the last several decades of the 20th century, the genetic code was broken by molecular biologists, and we present the genetic code as something like this to our students and to the world, okay? So that when we look at this, AUG, start here, A, first nucleotide, second nucleotide is U, so it's in this little quadrant here. The last one is G, that encodes a methionine or an M amino acid. CUU, C, first nucleotide, U, second nucleotide, third nucleotide is a U, encodes a leucine. G, here. Second nucleotide is a G and then a U encodes a glycine. Okay, and AUU would encode an isoleucine. You get the idea. And so then every amino acid in that 260 amino acid protein has a corresponding codon found in the mRNA, which has a corresponding codon in the DNA. And all of that goes through this decoding process. Okay, now go back to your Lotto ball machine and go back to your sticky note. You see where we're going with this. Intuitively, can we answer this question? Can design be detected in biological structures? Okay, I got one more. For you. Okay, so let's look at this one real quickly. This is super awesome, super fun stuff. So it turns out that there are machines in biological structures as well. Okay, and what you're looking at here is one of those machines, ATP synthase. Okay, that's what you see on the right. And ATP synthase does this. And you think, I'm not sure what that means over there. Okay, well, let's start with this. This here on the bottom is the production of ATP. ATP is the energy currency of the cell. It stands for adenosine triphosphate. Every time you move your muscles, you use a ton of ATP, okay? The end of metabolic processes, where we take in nutrients and we harvest energy from those nutrients, like carbohydrates, that in the end of it ends here at the production of ATP that then the cell uses as its energy source or energy currency. Okay. Now, this protein allows for the movement of protons. So this is a proton, or let's call it a hydrogen cation or hydrogen ion. This protein allows for the movement of hydrogen ions from a high concentration to a low concentration to make ATP. You need energy to make ATP, and this provides that energy. And you think, no, I don't know what you're talking about. That I don't get it. Okay, and if you don't get that, let's try this. Right, you are looking at a depiction of the Hoover Dam. Okay, and the Hoover Dam allows water to accumulate on one side, high water, and then has water on the lower side, right? On the other side, the low water, and allows water to drop from here to here through turbines that spin to then make electricity, right? They take the energy from the falling water and make electricity that then turns the light bulb from the off to the on, right? You get the idea. Okay, so that is what's happening here. High water, low water, need a turbine, okay? But that turbine, if it's spun correctly, can make ATP from this. This would be our light bulb off, our light bulb on, okay? That's what's going on here. This is the turbine. And you think, like, is it a turbine? What do you mean is it a turbine? Okay, is that just an analogy? Well, let's go here. I've got a video of this. This is pretty awesome, okay? So you are looking here at the turbine, right? Now look right here. This right here, okay, would be where there's lots of those protons or hydrogen ions. Okay, they're in yellow here, lots of them here. This is the Hoover Dam, okay, the concrete structure of the Hoover Dam. Here's where we have low water, okay, or low hydrogen ions, okay? And then this whole thing is gonna make the light bulb. It's kind of, right, the light bulb goes on to off. Actually, what's gonna happen here is it's gonna make ATP, which is shown here, from this green ADP, okay? And this is the turbine. Now watch this, okay? This is what actually happens. Hopefully this works. There we go, okay? Oops, we'll turn the sound off. There we go. Okay, so I'll kind of bring it back here. Right, so hydrogens are moving through this, causing this turbine to move, that then causing really cool biochemistry, molecular changes here that then allow the ATP to be made from the ADP. Okay, truly, it is a turbine. Okay, doing essentially what we see in the Hoover Dam. Now, what's interesting about that is that turbine that was shown, the video that I was showing is going pretty slowly. Okay, when it's running at full capacity, it can turn at about 7,000 RPMs. Okay, so let me show this. There we go. Let me show this video here. This is at probably real speed. Okay, there's two of them shown here, but watch the turbine here. All right, there we go, 7,000 RPM, somewhere around there, churning out ATP at a really high rate. This is a machine, right? And we see lots of things like this in the cell. So coming back to our question, can design be detected in biological structures? Well, let me quote some pretty well-known people and scientists. So this is a quote from Bruce Alberts. He is a former president of the National Academy of Sciences. He's written an entire textbook on cell biology. And he says this: the entire cell can be viewed as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines. Why do we call the large protein assemblies that underlie cell function protein machines? Precisely because, listen to this, like machines invented by humans to deal efficiently with the macroscopic world, these protein assemblies contain highly coordinated moving parts. In other words, Bruce Alberts looked at machines like ATP synthase and compares them to machines that have been invented by an intelligent being, an engineer, right? Now, go to Richard Dawkins. He says this: biology is the study of complicated things that have the appearance of having been designed with a purpose. Now, he'll go on to disagree that they've been designed with a purpose and they've been designed by an engineer. Okay, in fact, he'll write entire books on that. But the question is that because he's suppressing something that he intuitively knows, making a philosophical statement rather than a scientific statement? Okay, I think that the answer to this question and the conclusion that we can make is this: intuition would tell us that biological structures and processes exhibit features that allow us to conclude that they have been designed by an intelligent being. Okay, so, and I've given you examples of that, and I'm drawing on your intuitive innate ability to recognize that. Okay, now, last thing here. I know it's been a long day, and we got Jim Tour's talk coming up here in a little bit. Okay, I'm sorry if you keep hearing binging. I'm not sure exactly what that is, but anyway. So, but let me do one more thing here, and ask this question: should we trust our intuition? Okay, and Rob Statler actually talked a little bit about this, and Sal talked a little bit about this even as well. Okay, when we think about this, and I don't have a ton of time, so I'm going to go through this kind of quickly here, but should we trust our intuition? When we think about this, okay, when we think about what has to happen in the flow of evolution, if you will, okay, there has to be significant amounts of structural and functional innovation. Okay, so think about going from a single cell organism to an organism like you and I. There has to be significant amounts of structural and functional innovations that requires then significant amounts of molecular innovation. Okay, and Rob Stadler talked about at the end of his talk, he talked about like Richard Linsky and Sal did as well, and Gager's experiment on tryptophan synthase, showing that evolution doesn't really have the capacities to make new molecular innovation. Okay, that's what the empirical evidence seems to suggest. Okay, and he even talked about orphan genes, right? To start to get structural and molecular innovation, you have to have new genetic information, what they're called orphan genes, that have no recognizable similarities to any ancestral genes, which means they had to be made de novo. They had to come out of nowhere. Okay, so let me come back to this here. Okay, so this is our 260 amino acid protein. Okay, this is one sequence out of 10 to the 338 sequences. Okay, now I could change this methionine to something else and get the second of 10 to the 338 sequences of amino acids that, of this protein. Okay, or I'm sorry, not of this protein, but another sequence is what I'm trying to say. I'm trying to go too fast, sorry. But anyway, right, so when we look at this, the question is, okay, well, if we start changing these amino acids, are we going to get functional proteins out of this? And certainly we can. Okay, so there's not only one sequence that will lead to a functional protein. There's lots of sequences that will lead to a functional protein, okay, out of the 10 to 338. But there's also a lot of sequences in that sequence space that are not going to be functional. They're not going to have structure. They're not going to exhibit function. Okay, so then how many or how rare is it to get a 260 amino acid sequence of proteins that actually produces a protein? And Douglas Axe has answered that question, and I'm scaling his data, but he suggests that 1 out of every 10 to the 130 amino acid sequences will form a structure that produces a functional protein. In other words, we're going to have to search through this sequence space 10 to the 130 amino acid sequences to find a functional protein. Right? This is a big number. Does it present a problem, okay, for evolution? Well, let's do this. Let's assume that we'll start with a bacterial cell, okay? And let's say that we have 10 billion bacterial cells. And let's allow them to produce 20,000 generations a year. So in 1 year, we can have a total of 2 times 10 to the 14th bacterial cells. It's a lot of cells, okay? Every single one of those cells can sample a new sequence, okay? Let's give them 5 billion years to do it, okay? How many sequences could those cells search? They can search about 7 in 10 to the 23rd sequences, right? Now, how many do they have to search? 10 to the 130th to get 1 gene. 1 gene, okay? Now, here's the problem. Take your simplest cell, okay? This is our minimal cell, a cell containing the minimum number of genes needed to carry out functions necessary to sustain life, okay? This simple cell, okay, has 473 genes encased in 531,000 nucleotides, about. Compare that to us. We have 20,000 genes encased in 3 billion bases, 20,000, 25,000 genes. Okay, so when you think about the evolutionary paradigm, we have to go from here, okay, and at 473 genes, and there has to be 20,000 plus genes made, okay, encased in billions of genes, right? And I'm saying that this is going to have a hard time just making one, and there has to be 20,000 that are made, right? So should we trust our intuition that we can recognize design in biology? I think we should. Okay, I think that it leads us to the correct conclusion, and Romans 1 tells us that it will. Okay, all right, so that's where I'll end my talk. I know the last part of that I went really fast. Maybe we can catch up on that in the Q&A, but I wanted to give you some time to ask some questions now. Plus, I want to make sure that we keep in schedule for Jim Tour's talk as well. All right, thank you for your attention.
Awesome,
Any
thank
questions?
you Thank you so much, Dr. Sholders. Yes, there's several questions here. We'll probably only get to 1 or 2, but we'll save the rest for our panel discussion. So the first one is from Solidify Your Faith. Is water a problem for the origin of life?
I can't hear you. I don't know.
Oh, let me see if I can fix this.
We can hear you. We can hear you loud and clear, Crystal.
You're good?
Yeah, it's probably on his end somehow. He may have to.
I'm not hearing anything. Maybe I lost something. Maybe I'll take off my
maybe his earbuds. Yep.
Okay. Can you hear me now?
Can people hear me?
Yeah, we can hear you.
We can hear you loud and clear.
I can hear you now. Okay, good. Sorry.
Perfect.
Okay.
No worries at all. We do have several questions here, but we'll save the rest of them for the Q&A. If you're sticking around for that, that would be absolutely wonderful. We will take a few here though. So Solidify Your Faith asked, is water a problem for the origin of life? Does hydrolysis prevent molecules from joining together to make life in water?
Oh, that's an interesting question. So is water a problem for life? Does it prevent molecules from coming together? So water is used to break many of the bonds that hold macromolecules together. However, the macromolecules, when they are held together, okay, are fairly resistant to being broken up by water, and you typically need some sort of enzyme to catalyze the reaction, okay? But from an abiogenesis standpoint, and this is where I'll probably leave Jim Tour to that, water could cause problems, okay? And so, but that's really not my field of expertise. So that's a much better question to actually ask Jim Tour because he's an organic chemist. So great question, interesting question, one that certainly needs to be talked about and thought of. In particular, when you think about abiogenesis, which is essentially making macromolecules without all the cellular apparatus to do so, the transcription translation processes that I talked about.
Awesome. I think we have time for one more. Can amino acids used for life form naturally? Can they join together to make proteins naturally? And can proteins join together to make a living cell naturally?
Great. Again, that's actually an abiogenesis question. So when we think about it from a cellular standpoint, we have in our cells things that can make amino acids from raw materials. We have things in our cells that can make peptide bonds. Those things are enzymes. Those things are, other, like RNA machinery, those kinds of things, all sorts of things that allow for the production of peptide bonds, that allow for the production of amino acids, okay? So biologically speaking, yes, we can form amino acids. Now, if you're asking the question without the cell, then you're talking about A, without, bio, right, the cell, biogenesis, the production of something, and I hope that you'll see answers to that question with Jim Tour. I think that he'll probably point you to the fact that there's going to be major difficulties with that. Okay, that these things are not just going to come about naturally given the conditions that are supposed to be there in the prebiotic soup. I don't want to steal all his thunder, but that's the majority of what his talk is going to be about, and certainly what his field is about as well.
Awesome. Thank you so much for your time and the wonderful presentation, Dr. Sholders.
Yeah, I'll
really
stop.
appreciate it.
The giraffe that sings. A while ago, a fellow asked us in an email why in the giraffe a nerve descends all the way to the bottom of the neck, then goes up to the other side to the larynx. Isn't that evidence that God is a poor designer? I replied, when you wire a house, do you go around the walls or do you send it the most direct route diagonally across the room? Apparently, you must be a much better designer than God, so I'll give you an experiment. Go into the giraffe and surgically shorten the left recurrent laryngeal nerve. Perhaps then the giraffe would be able to sing. Speculating on how you could design something better than God is arrogant and quite foolish.